All right. Good afternoon, everybody. We're going to start our meeting today. Assalamualaikum warahmatullahi wabarakatuh. Praise be to Allah and salawat to Rasulullah sallallahu alaihi wasallam. Today we are going to have an invited speaker, Mr. Khizar Ahmed Suman, Professor Suman. He is from Bangladesh. He is from Department of Fisheries Management, Bangladesh Agricultural University. My men Sikh, Bangladesh, is that correct, Professor Suman? I can't hear you. You mute. You have to unmute your speaker.
Yeah. Now can you hear?
Oh, please unmute yours. Yeah.
Yeah. Now can you hear me?
No. Yeah. Clear. All right. Thank you. What time is that? What time in your country?
Is my video okay?
Okay. So I think in your country is too late right now, or what? Too early in the morning, or what?
Actually, it's just beginning of the afternoon.
Oh, same time like in Indonesia. So we are at 1:30 right now, actually.
Yeah.
So
yeah.
I just want to read your CV. You, Professor Suman, is in the position of the Associate Professor at Department of Fisheries Management, Bangladesh Agriculture University. My Mensih, Bangladesh, is that correct? I don't know. Forgive me for my pronunciation of your. Okay. So he got some awards, 5 here. So Best Publication Award 2020 by Bangladesh Agriculture University Teacher Association. They call it BUTA, something like that. I don't know. And also Best Publication 2019, 2018. And then Netherlands University Foundation for International Cooperation, NUFIC Fellowship 2014 to 2018. And National Science and Technology NST Fellowship for 2012-2013. So his Professor Suman interest is ecotoxicology, ecological risk assessment, stress ecology, and pesticide toxicity, fish toxicity, microplastic contamination, climate change impacts. So he already works for 20 papers to be reviewed. So some of them are environment, international chemosphere, environmental science, and pollution research. So some of his publication you can search. Everybody can search for the professor publication in the Google Scholar or Google something like that. So we don't want to waste our time. Professor, you have here the schedule. You have 10 minutes for your presentation. So the time is yours. Thank you.
Can you see my PowerPoint, please?
Yeah, clear.
Thank you. Respected session chair, distinguished participants, ladies and gentlemen, very good afternoon, everyone, and welcome to my talk. I am Dr. Khizar Ahmed Suman from Bangladesh Agriculture University. Already session chair introduced me, so I'm not going to introduce myself. Very welcome to my talk. I'm going to talk about pesticides in aquatic environment in Bangladesh, assessing impact using freshwater fish models. If we look at this graph, we see the global pesticide production and imports in the last decades has been increased sharply. And the similar trend was observed in Bangladesh, actually. Here we see from this graph, about 500% of the increased use of pesticide is observed in last 2 decades. Bangladesh has ranked the 2nd position in terms of the increased use of pesticide just after Argentina in last 2 decades. From this graph, we see that Rahman showed that about 15 times of the use of pesticide was observed in Bangladesh in just 4 decades. So, what is the motivation of the use of the pesticide in Bangladesh, actually? What are the reasons behind the increasing use of pesticide in Bangladesh? We all know Bangladesh is one of the most densely populated countries over the world. About 160 million people are residing in Bangladesh. So due to the habitat construction of the ever-increasing population, the available arable land is decreasing day by day. At the same time, several agroclimatic variables like seasonal flood, flash flood, cyclone, storm, salinity intrusion in the coastal belt pose further difficulties to agriculture crop production. So if we want to meet the ever-increasing population, there is no way except the agriculture intensification. And considering the agriculture intensification, we need to produce more high-yielding varieties, which is mostly susceptible to disease and pests. So farmers use the pesticide indiscriminately to protect their crops from the pests. And these pesticides are eventually find their way into the aquatic systems through several exposure routes. If we see this picture, we see the spray drift scenario, spray drift and surface runoff, groundwater seepage are the major routes of the pesticide into the aquatic systems. Apart from that, the equipment washing water, when the farmers use pesticides in their crop field, just after using the pesticide, they wash their equipment in the nearby aquatic systems, and this is the one of the major routes. And sometimes, due to the cheaper of the plastic bottle or pesticide bottle, they just throw away, dump away into the aquatic systems, and this is another way of the pesticide residues into the aquatic systems. And when the pesticides are into the system, aquatic system, the different trophic levels like primary producers, Diopnia, invertebrates, and other fish species, different fish species are getting contaminated from the pesticide concentrations. So what I did in the last couple of years about the pesticide research, actually I did my PhD in Bangladesh, in the context of Bangladesh. I got my degree from Wageningen University in the Netherlands with the ecotoxicology group. I had a field survey in 2016 about the inventory of pesticides and the risk assessment in different organisms. We conducted chemical monitoring about the organophosphate groups of this pesticide residue analysis and the risk assessment for different organisms. I had microcosm experiment and several laboratory experiment with fish. Chlorpyrifos, Phenetration, Sapamethine, Dacloprid, they're the major groups, major pesticides which are used in Bangladesh. So now I'm going to talk about 2 recent investigations of mine, but already this paper are submitted in different journals actually. I'd like to talk about the thiamethoxam pesticide, insecticide, which is the neonicotinoid insecticide, on the developmental toxicity of banded gourami, which is the popular fish in Bangladesh, India, Pakistan, etc. We investigated the hatching rate. The hatching success was decreased significantly with the progression of the thiamethoxam concentration. And the mortality of embryo and larvae increased, incubation period was decreased. We just after fertilization, we exposed the fertilized eggs were exposed to the different concentrations of thiamethoxam. We see some several malformations, unhealthy embryo, dark brown, yolk sac, eggshell broken, yolk sac edema, etc. So among the abnormalities, morphological abnormalities, yolk sac edema was the most the observed malformation. And for the one-day-old larvae, we also saw some malformation exposed to the thiamethoxam pesticide. Here we see notochordal abnormalities, irregular caudal fin or incomplete caudal fin, yoxagadema. Here we see lordosis, and so on. So this is all about, yeah. And for the frequency, if we see, consider the frequency, here we also say that yoxagadema was highest observed malformation in one-day-old larvae of banded flora. So this is all about the developmental toxicity of thiamethoxam on the banded flora. Now I'm going to talk about one of the important pesticide, profenophos, which is organophosphate groups of pesticide, and which is extensively used pesticide in Bangladesh based on our previous survey. On the Nile tilapia, you all know about Nile tilapia. It's a ecotoxicological study animal, and it's efficacious distribution in nature, short generation time. For this, why we selected this fish. Firstly, we conducted medium lethal concentration test, LCT test, and after that, based on the 96 or LC50 test, we conducted another experiment to know the toxic effects of profenophos on hematological parameters and some blood biomarker study, and also some histopathological observations. Firstly, for the medium lethal toxicity test, we followed OECD guidelines, which is the standard protocol for LC50 test. We used 9 concentrations using duplicated treatment for this experiment. Using probability analysis, we calculated the 96 LC50 of profenophos for Nile tilapia is 1,614 microgram per liter. And based on this 96 LC50, we had another experiment. 10 male fish of Nile tilapia was exposed to the 5 sublethal concentrations, 0, 5, 10, 20, and 40% of the 96 LC50. We had 3 replications. Pesticide was applied 7 days interval. About 90% water was changed, and new profenophos concentration was used. And this experiment was lasted for 28 days. It was chronic experiment actually. We measured blood glucose, hemoglobin, WBC, RBC using standard protocol, and some histopathological observation using standard protocol. Glucose level was increased with the progression of the profenophos concentrations. And opposite scenario was observed for the hemoglobin levels. It was decreased significantly. And for the RBC level, it's also same, almost significant decrease. And for the WBC levels, it's also significantly increased with the increase of the profenophos concentrations for different sampling days. We studied the erythrocytic cell nuclear abnormalities using blood smearing. Here we see some nuclear degeneration, micronucleus, binucleus, nuclear bud, karyopetenesis, etc. And for the cellular abnormalities, we also see some abnormalities, of course. This is teardrop, elongated cellular cell, and some twin and spindle-shaped cell in nucleus in erythrocytes also observed. And for this pathological alteration of testis of Nile tilapia, we see some alteration of empty lumen, degeneration of Sertoli cell, breakers of seminiferous tubules, etc. And for the liver histopathology, we see some necrosis, hemorrhoids, pyknosis for different sampling days. And also for the kidney, we also see some alteration like necrosis, hemorrhoids, pyknosis, etc. Vacuoles, here is some vacuoles. And for the gill histopathology, we see some clubbing, telangiectasia, necrosis, etc. So this is all about the effects of the profenophos on the Nile tilapia blood biomarkers and histopathology. So this is the last slide of my presentation is how to reduce the pesticide concentration actually. In Bangladesh, I don't know about the Indonesia. In Bangladesh, we know that farmers use actually pesticide indiscriminately. They're just very much illiterate. They don't know about the pesticide level and sometimes don't know the doses of pesticide, which is prescribed by the agriculture officer. They just use more and more pesticide. That's why we need to educate the farmers on the proper use of the pesticide use. And another alternative method, I don't know whether it's very popular in different countries, but in Bangladesh, it's not yet popular. It is IPM method, integrated pest management method. And we need more research on the biopesticide, how to use alternatively against the synthetic pesticide. So this is all about my talk. Thank you very much for patient sharing. If you have any question, please feel free to ask. Thank you.
Sorry, I forgot to unmute mine. Thank you very much, Professor Suman. You have 5 more minutes, so we can have some discussions of this one. I think it's very interesting. If anybody in this forum have question, please ask to Professor Suman because he has some answers on this one. Very interesting topic actually. So if anyone have question, please raise your hand or just talk and unmute your mic. No one? Okay.
I think Ilham Fahmi raised his hand.
Yeah. Okay. Okay. Please welcome Ibu.
No, I think Pak Ilham raised his hand.
What? Oh, Pak Ilham. Yeah. Anybody who raises, I can't see it. So you can talk. You start talking and then ask Professor Suman.
Thanks, Professor Muhammad Ilham. Nice to you meet.
Hi.
You back in this virtual room. I hope you are doing well here. Very interesting presentation, I thought. But I want to ask you, I think that is a very interesting question. How do we distinguish the negative effect of the pesticide with or compared to various other pollutants in the water ecosystem? As we know, so many pollution in aquatic ecosystem. How to distinguish pesticide with the others pollutant? Thanks for the explanation. Keep safe and stay healthy.
Yeah, thanks for your question. Actually, we can assess the negative impacts of pesticide using different methods. For example, from my PhD dissertation, I had one chapter I published in Chemistry Journal. We assessed the pesticide residue in different water bodies in Bangladesh. We just collected the sample, water and sediment sample, and analyzed the residue of organophosphate groups of pesticide using GCMS. Then we analyze the residue or concentration of pesticide, and we also assess the risk actually using risk quotient method. So for the primary producers, invertebrates, and fish, model species, of course, there. So when the risk quotient RQ value was greater than 1, then we can say that, yeah, it's a moderate or moderate risk for these organisms. And when it exceeds to the RQ value exceeds the 100, then we can say it's severe risk for this organism, particular organisms. So from the open water system, if we analyze the pesticide residue and assess the risk, then we can say, yeah, pesticide has the negative impacts on the aquatic systems. Are you clear or?
Okay.
Is it okay?
Thank you very much for your explanation, Professor Tiza.
Yeah, thank you.
All right, thank you, Dr. Ilham. Anybody else, please?
I want to take a picture actually of this, if you don't mind.
Right, fine. So while you are taking pictures, I'm gonna let somebody else asking some questions. If not, I'm very interested in your topic actually. So it's not part of my focus, but part of your subject is part of my subject to environments. So I like to have that one. So the pesticide inland is not only a problem in your country, also a problem in our country. So I think all over the world. So is there any way that we can do it to catch up the runoff from the runoff the water, to have a small basin or something like this, and then at that small basin we do the bioremediation to clear the water, and then water can go through to the outlet such as a small stream or rivers, and then we can have we are happy if anybody want to have a pesticide in their land because of the population is growing, so everybody needs food. So food to be fertilized with the biofertilizer is very hard to find right now. So they do the cheapest way is by doing the pesticides. So when the runoff occur, then we have to catch up the runoff and build a small basin, and then we treat the small basin being good water cleans, and then go out like to the water to the river or stream. Is that any idea of this one? I don't know. I think I have an idea like this. So what do you think, Professor Suman?
Actually, what is your main question? I can't get it. Maybe
my question is, can we do the water treatment before it go to outlet?
Actually, I don't know. It's very, very difficult, I think, because when runoff occurs or spray drift occurs, for the spray drift scenario, you can use the spray drift buffer. I can say that some developed countries, they are using this technique, spray drift buffer. So in between the agricultural land and the nearby aquatic system, they have a very high tree. They plant very high tree. So there is minimum or a considerable percentage of the spray drift are stopped, are hindered by this tree. So we say it's a spray drift buffer. For spray drift scenarios, I have only a suggestion, or I have only a proposal, but for the runoff, I don't know. It's very tough to maintain this because, yeah,
Yeah, understandable.
The only thing is we have to reduce the pesticide application in crop field by motivating the farmers, I think. Because
everybody wants to do that.
Yeah, because they are very much illiterate. They don't know how much doses they need to use. Sometimes they use, in the most of cases, just they use overdoses. That's why this is the problem occurring actually. Yeah?
All right. Thank you, Professor Simmons. We have exactly on time right now. Everybody, please give applause to Professor Suman. Thank you very much.
So thank you very much for patience hearing, and thank you for inviting me to talk something about my research. So thank you.
You are welcome. For the next speaker will be Umi Muawana. Are you here, Umi Muawana? So we gonna give you 10 minutes presentation.
Sure, thanks.
So if you can present for 7 minutes and then 3 minutes for discussion, that is very good. But if you run out of the time, 10-minute presentation, so you don't have any more chance to have a discussion with us. Thank you. So time is yours.
Thank you. My name is Umi, and I'm researcher at the Ministry of Marine Affairs and Fisheries, and I'm an economist. So today, I would like to share one of our findings on the seaweed farmers. Actually, one of the interesting thing is only like one small, unique and important segments of seaweed farmers. So we are not talking like in general seaweed farmer, but this is more on fishers who capture seaweed. Are they fishermen or are they farmers? So yeah, I would like to share that. Well,
would you have a full screen, please?
Okay, sure. Okay, good, thanks. So yeah, seaweed is one of top or important priorities development in Indonesia and in MMAF, Marine Affairs and Fisheries Department here. That's why it's one of an interesting topic to look at. And today I would like to share how the seaweed farmers' economics and livelihood can play an important role on poverty eradication over there, particularly in North Kalimantan. It's in Nunukan. And also some aspect of seaweed fishers and, well, seaweed fishermen. And also other things relates to conflicts among them. What are things triggering conflicts and what are the resolution taken? And from there, we would like to recommend methods for seaweed farming development in Nunukan. Well, Nunukan is, oh, I don't have the map. Well, here, Nunukan is actually a district in North Kalimantan province, and it's really adjacent to the Malaysia. It's next to Sebatik, and then we have Malaysia on the north part bordering with us. So that's where the location of the research. And we have seen here that Nunukan district is one of the source of seaweed throughout Indonesia, as well as being exported to Malaysia. And this is some of the findings. So the finding we will explain like in terms of their economic performance, the asset ownership of the seaweed farmers. So in general, I will not read up all of them, but let me try to bring you to vision or imagine looking at the coastal communities of seaweed farmers. In general, they all have their own areas or farms, which is on the sea, of course. And then they have different set of asset ownership for production. So the basic characteristic, like they have around 0.5 hectares of their farming sea, or well, they don't have land. So, and then one of the asset that they have on land is actually drying areas. That's when you see a seaweed farmer have drying areas, that means they are a little bit in a better economic condition than others who do not have any. So that's one of the critical point. So the average asset value that they have is not like way too high. It's 16 million for asset. And then if you look at, and this is the number of the the farmers, like 700 or something. It's about 2,000 in the past, but then decrease again. But if we see the average income, it's around like 5 million, and then 7 million. So basically, if we compare with minimum wages, which is around 3 million, I guess. So sort of at glance, they are in a better economic condition. And if we look at deeper on the social part of their origin, of their histories, many of them are actually migrants or leaving the migrant condition in Tawau or Malaysia and then become seaweed farmers in Nunukan. In other words, this business is actually helping them economically, helping them to live out from poverty, things like that. So and among one or two of them have really, really like huge asset, like in terms of really nice housing. So, but that's not in general. So this is in general their condition. And if we see their expenditure, daily expenditure, well, they spend food quite, well, they spend food like 50%. In economic theory, usually we spend food 30%, but if you spend more than 50% of your income for food, then not super wealthy. But that's their condition, and usually in small islands on the outermost of Indonesia, everything is so expensive. I think that contributes to their welfare, I think. And then we have all the dine out cigarettes and everything. Based on their expenditure, is like around 4.4 million, and we see their income is way more than that in when we saw previously. It's above their UMR or minimum wages in that region. And the second one is the one that like around 700 people over there are actually fishermen capturing seaweed. So this is particularly interesting, only happening in Nunukan, and they are all small boat owners. And when the price of the fish is not really good, and they switch to seaweed fishes, I call it. And this has become a debate locally in the local government, whether they can be categorized into farmers, seaweed farmers, or fishermen, because they have all the fishing gears, but they don't have the farms, but they only capture the seaweed and sell the seaweed and then dry the seaweed and everything. So this is the thing that I would like to share on top of the economic condition of the seaweed farmers. So they have better price. They catch like as big as their boats can get. So it's about 1.5 tons of wet seaweed per day, and they usually capture them around the farming area, the farming centers, because a lot of currents over there, and they capture the things that was run off by the currents. But that also triggers some conflicts. One is conflict among the seaweed farmers themselves, claiming areas of their own, because all of them are public goods, public sea areas. So sometimes they have some issues with neighboring farmers on because some of them are expanding their seaweed farming areas. And then some conflict between the seaweed farmers and the seaweed fishers, I call it. Because sometimes, well, we don't know exactly the quantities, the frequencies, but occasionally it happened like either the fishermen cut the ropes, or the farmers think that their ropes of the seaweed farming being cut by the fishermen who waiting for the seaweed. So those are the things happening there, and it has drawn a lot of attention from the local government, namely fisheries officers in Nunukan. And many of them has been resolved through formal and informal meetings, like not being used.
You have 1 more minute.
Okay, 1 more minute. So yes, and the things that we recommend here, because seaweed is a good for livelihood. So long-term development plan needs to really not navigate, dedicate areas and enforce them for seaweed, for the farming areas. And then there is also a provincial government regulation over there, number 4, 2014, actually already zoning the areas of North Kalimantan province. But in reality, the implementation, the socialization, and everything is still weak. That's why sometimes creates conflict among the fisheries or marine users, including seaweed farmers. Okay, so that's all. Thank you.
Thank you very much. Unfortunately, we don't have any time to discuss about the issues. Might be later on if you stay in our room, and then we have some time so we can talk about it, because interestingly, some seaweed is losing from their planting, and then somebody catching and then fighting for that one. All right, thank you, Ibu Umi Muawana.
Yes, thanks.
I'm not so good for this one. Get applause to Ibu Umi.
Thank you.
All right, for the second speaker will be Jacqueline M.F. Sahetapi from Upati University. So room is yours. Welcome.
Thank you, moderator. Assalamualaikum warahmatullahi wabarakatuh. My name is Jacqueline Sahetapi from Patimura University. Today, I will talk about hematological response of juvenile tiger grouper to toxicity of heavy metal lead.
We have some issue on, showing your presentation because of the technical problem.
Oh, yeah.
Okay, can you?
Yes.
Do you have your own presentation or no? Oh, okay. Ini lagi, Ibu. Okay, you can start right now, Ibu Jacqueline. You have 10 minutes.
Sorry, it's a problem.
You can start your presentation.
Yeah, yes. Okay, I'm so sorry. Today I will talk about the hematological response of juvenile tiger grouper to toxicity of heavy metals lead. Heavy metals are produced from different sources, including natural and anthropogenic sources. Heavy metal contamination of the aquatic environment results from direct atmospheric deposition or geological weathering or through the evacuation of agriculture, municipal, residential, and manufacturing waste products, as well as via agriculture wastewater treatment. Gracia 2015 alleged that
So do you have technical?
Sorry.
Bisa.
Next, the introduction, sorry. Yes, of course. Okay. Naturally, lead enters the waters through the lead crystallization in air under the auspice of rain, leaf of fine grains of lead, and also the fuel contained of tetraethyl lead, which contributes to the existence of lead in water, erosion, corroding process of mineral stone, and industrial sewage such as factory of battery, ammunition wire, and paint. Lead compound that enter the ecosystem, becoming the source of contamination and affecting the life of aquatic organisms. For example, the high concentration of lead toxicity can be lethal to fish, particularly at larval and juvenile phase. Lead and its compounds are extremely toxic and pose a major threat to the animals and humans' health. The human intoxication by lead is usually conducted by eating poison marine organisms and polluted air. Some research have shown the existence of certain heavy metal in high concentration shall affect the toxicity to fish. Based on those perceptions, it is important to conduct a research to analyze the effect of lead toxicity to hematological response and survival rate of juvenile tiger grouper. The purpose and benefit of research was aimed to analyze the effect of some concentration of lead toxicity to hematological response, including concentration of hematocrit, hemoglobin, number of erythrocytes and leukocytes, and also survival rate of juvenile tiger groupers. This research was expected to provide information about the effect of some concentration of lead toxicity to hematological response and survival rate of juvenile tiger grouper. Next. Material and methods. Material used in this research were juvenile tiger groupers, a sample the size of 6 till 7 centimeters obtained from Situbondo hatchery, marine water as a culture media, pellets as food, and heavy metals. Lead, which used in form of nitrate lead. Tools used in this research are aquariums, refractometer, diometer, pH meter, magnetic stirrer, and measuring cup volume 500 milliliters. The research are divided into 3 steps: concentration tests, acute tests, and subchronic tests. The tests are held in 3 treatments and 3 repetitions we described as following. Next. The effects of some concentration of lead toxicity to hematological response and survival rate of juvenile tiger grouper are analyzed by processing data using analysis of variance, then continue with 2K tests whether the result provide the real impact. Supporting factors are analyzed with descriptive statistics in form of table, graphic, and figure. Next. The results or data of hematocrit concentration shows that there is reduction of hematocrit concentration at all treatments until the measurement at 30 days, where the higher lead concentration is significantly able to reduce the number of hematocrit in blood of juvenile tiger grouper. The next. The next figure shows that after 30 days exposure, the further effect of lead toxicity becomes significant at 10th day, of exposure at treatment of concentration 6.86 ppm, which reduced the hemoglobin concentration in blood of juvenile tiger grouper. Next. The figure show that after 30-day exposure, the further effect of lead toxicity begins at 10th day of exposure at treatment of concentration 6.86 ppm, which reduced the number of erythrocytes in blood of juvenile tiger grouper. Next. The figure of leukocytes show that after 30 days exposure, the further effect of lead toxicity begins at 10th day of exposure at treatment of concentration 6.86 ppm, which increased the number of leukocytes in blood of juvenile tiger grouper. Next. And the last, survival rate. This figure shows that the declination of survival rate percentage at all treatments. The results suggested that the higher lead concentration will reduce the percentage of survival rate of juvenile tiger grouper due to the damage of its body tissue, particularly the gill, that led to the reduction of gill's ability in absorbing oxygen, thus causing anemia and death to fish. Next. The conclusion, based on the results, some conclusion may be suggested as following. The highest effect of lead toxicity on tiger grouper reduced concentration of hematocrit and hemoglobin 2.64% and number of erythrocyte 0.77 times 10 raised to the power of 6 cell per millimeters at 10th day of exposure at lead concentration of 6.86 ppm, while the number of leukocytes is increasing up to 0.81 times 10 raised to the power of 6 cells per millimeter. The toxicity of lead affects the survival rate of juvenile tiger grouper, of which the higher lead concentration will reduce survival rate. Okay, I think that's all. Thank you.
Thank you, Ibu Jacqueline. We have 1 more minute to discuss your finding. So if anyone have a question, please raise your hand or just talk because I can see. Can you close the? Yeah. Okay, anyone have questions so we can discuss about it? If you don't have, we're gonna move to the next speaker. It will be Professor Nur Fadli. Are you here, Professor?
I'm here, Mr.
Oh,
Irham.
You are part of Professor Nur Fadli. Okay, so you have 10 minutes for your presentation. So time is yours and welcome.
Okay, thank you, Mr. Muhammad Irham. Assalamualaikum warahmatullahi wabarakatuh. Thank you for time that you to me. Let me introduce myself. My name is Nanda Muhammad Razi. I'm student from Universitas Syah Kuala. In this opportunity, I would like to present about my team research by the title Checklist of Commercially Important Grouper Epinepelidae in Aceh, Indonesia. Okay, next. Next. The grouper, locally known as kerapu in Indonesia, is one of commercially important fish in Aceh as well as many other regions and countries. Worldwide, the Serranidae consists of the last 160 species belong to 15 genera. In Indonesia water, at last 77 species of Serranidae have to be recorded, include the recently described species Epinepelus cupangenesis. Next. An accurate species list is one of the most important information for strategizing an optimal fisheries management plan. Unfortunately, in Indonesian fish landing site, members of group of collectively record as crapo instead of the precise species name. Okay, next. The present study was aimed to catalog the group of species that are landed in Aceh fish landing site, providing baseline data that will be valuable in Aceh fisheries management plan in the future. Okay, next. Time sampling on 2060 and 2021, location in Sabang, Aceh Besar, Sigli, Los Mawe, Idi, Langsa, Aceh Tamiang, Calang, Melabuh, Tapak Tuan, Semelu, and Singkil. Okay, next. Species identification. The sample we are identified based on Hemstra and Randall's 1993, Frost and Pauli 20 and 70, FRCA 2020, record and photograph. Okay, next. To see conservation status, we use IUCN and CITES. Next. This IUCN and next. This CITES web. Okay, next. For data analysis, data of local distribution was analyzed according to Muklisin and Siti Azizah 2009. Next. The distribution and species composition, 41 species of Serenidae family we are identified. Epinephelus was the dominant genus found in Aceh, 56%, followed by Cephalopolis 29%, Variola 5%, Pretopomus 5%, Aetaloperca 3%, and Aniperodon 2%. Next. The red box identification height species variant in the location. Okay, next. From the red box, this indicates some species have high distribution value in many locations. Okay, next. For conservation and trade status, we have 88% less concern, 5% data deficit, and 7% vulnerable. Okay, next. From this, the red box shows some species are decreasing in population. Okay, next. Okay, the conclusion is in this study, 41 species of Serenidae family were identified. Epinephelus areolatus had a highly local commercial distribution, 92.31%, followed by Variola albimarginata, 69.23%, Cephalopolis soneratia, 46.15%, Cephalopolis buena, 46.15%, and Epinephelus coides, same. Epinephelus tucula had the lower distribution in Aceh, 7.69%. Third, 36 species for 88%. We are categorized at last concern to species data deficient. Will 3 species we are to be vulnerable category. The result of the study has implication on the sustainable fisheries management, the regulation of at the provincial level protect the species from overfishing. Okay, next. Okay, thank you very much for we all know it's one no perfect. I think my English is broken. I hope the presentation easy to understand. Wabillahi taufiq wal hidayah. Assalamualaikum warahmatullahi wabarakatuh.
Thank you very much, Mr. Nanda. Give him applause for his presentation. We have 1 more minute to have some questions, some discussion. If anybody have questions, please raise your hand and then talk. Anybody, please? Okay, if you don't have any, we're gonna let other speaker to have a talk. The next speaker will be Professor Muhammadar. Are you here, Prof. Muhammadar? Or anybody represent Prof. Muhammadar? No? Professor Muhammadar should have 2 talks today about shrimp biology and study of amino acid. So if might be Professor Muhammadar have another busy job, we're gonna let anybody first, and then we're gonna let Prof. Muhammadar later on. The next speaker will be Bapak Lukman from Research Center for Limnology Indonesian Institute of Science.
Yes, Pak Irham, I represent Pak Lukman.
Okay, you present for Pak Lukman. Okay, you have 10 minutes for presentation, so time is yours. Welcome.
Okay, would you please show the presentation? Okay, Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone. I'm Octavianto Samir from Research Center for Limnology, Indonesian Institute of Sciences. I'm here represent Dr. Lukman, Dr. Trianto, Professor Gadis from LIPI, and also Ms. Gogali and Mr. Banjolu from Mesintu Institute Poso. Okay, I would like to share our research titled Eel Fishing Activity in Poso Area, Central Sulawesi, Indonesia. Next. Okay, this is my outline presentation. Maybe we can skip that. Next. Okay, Indonesian waters are hypothesized as the ancestor of Anguilla's area because we can find 7 from 18 global species or subspecies in Indonesian waters. One of the eel production areas in Indonesia is in Poso. In Poso waters, we can find 5 species of Anguilla. The highest abundance of leptocephali, as we know as larva of anguillids, is also found in Poso, in Tomini Bay. Poso eel production reached 22 to 54 tons per year in 1970s. And in 1990 and 1998, the eel production in Poso was around 41 and 30 tons respectively. No wonder if Poso being one of the main areas of anguillids production in Indonesia. From those descriptions, we can assume that research on the fishing activity of eels in Poso is highly required for evaluating and determining policies that affect the caught of anguillis. And next. The information that we had in this research comes from observation, interviews, and secondary data. The observation itself, which results in primary data, focuses on caught by wire and spears from April to June 20 and 21. Next. Now I want to share about the result of this research. From the picture, we can see that the fishing activities are spread over the lake, mainly in the shore areas, Poso River, and other rivers as the lake's inlets. The fishing gear commonly used were poles and lines. These gears are used in order to fit with area condition, which is dominated by rivers. Trap also found in the inlets and also the outlets, Saojo. The trap that local people use is called bubu, a trap made from bamboo. We go to another fishing gear. Another widely used gear is a 5/8 spear with 5 to 7 meter long bamboo stick. Fishing using spears is a traditional activity of the local people known as monyelo. This activity is generally conducted in shallow water area, especially in Poso River, around the outlet of Lake Poso, and on the shore of Lake Poso itself. Next. Okay, another fishing gear, which is the main fishing gear in Poso, is called waya, or a fence system trap. Waya is kind of large trap that's sized 20 meters in length. The mouth of waya is about 5 meters in length and toward the water flow, as shown in the picture. Similar to fence, bamboo is arranged as the side of waya. There is a 1 meter square rectangular trap in the end part of waya. And this waya is distributed in the Poso River around the outlet of Lake Poso to catch eels, which downstream migrate to the sea. Next. Okay, from the graph, we can see that the eel production from Poso in the last 4 years was around 6 to 15 tons per year. This was above the eel production in 2004 to 2009, which was 5 to 8 tons per year. And the next graph shows that shows the eel count per month from 2004 to 2006. From this graph, we can see that the eel's production fluctuated seasonally, with its high production occurred in April, May, and June. In the last month of the year, the eel production decreased, and in the August, October, and November, there was no catch of eel. Fluctuation of eel production was related to the water level in Lake Poso. Based on the data from Poso Energy, the water level of Lake Poso was quite high from April to July. Other research from Sugeha also said that the monthly catch of eels migrating downstream from Lake Poso would increase when the water level was high. Okay, next. Okay, this graph shows the Poso eels production from 2017 to 2020 in 5 areas in Poso. The highest productivity is in Pamona Pusalemba, ranging from around 5 to 13 tons per year. This area is the central location for waya fishing. If we compare to the current data, which taken in March to June 20 and 21, the catches of eels in Poso were in range of above 1 ton per month. The catches from waya and spears were only a bit different. The weight of eels caught was ranging from 8 ounces to 10 kilograms, with the length in range of 68 to 135 centimeters. Okay, next. We go to the evaluation of this research. The Poso waters which consists of Lake Poso, Poso River, River Estuary, and the Tomini Bay, is a complex and perfect mixed habitat for supporting the development of eel. If we talk about the species of Anguilla, the dominant species is Anguilla marmorata because it can live in various habitats and high elevation area. This species inhabits and undergoes a phase of life from seawater or brackish estuaries to upstream rivers and lakes. Talk about the fishing gear. Local people use traditional and specific fishing gears such as wire and spears. Like I described before, wire is indicated as the unfriendly environmental gear because it's blocked the downstream migration of the eel. And also the eel size caught was big size. It's more than 70 centimeters in length. As we know that eels that go downstream migration with more than 70 centimeters have already developed gonads. Okay, fortunately, the eel fishery is not threatened by caught fishing because nowadays wire much more decreased. And the second is other fishing gears passively operated. On the other hand, habitat changes play a more critical role in eel treatment. For example, the first is changes in the aquatic terrestrial transition zone or ATTZ. For example, wetland, the changes between wetland to farm. And the second one is the dam, which can obstruct the eel migration. For this case, for the dam case, I mean, a fishway can be a solution, although the effectiveness should be tested to make sure the eel migration was successful or not. Okay, next. From this research, we can conclude that Poso water, included like Poso and Poso River, are the main eel fishing areas in Indonesia based on their location and habitat complexity. The second is Waya and Monyilo, or we can, as we know as spears, which local people use, highly contributed to the eel catch fishing in Poso. Next is the sustainability of eel fisheries in Poso will be threatened, especially by habitat change, such as the reduction of TTZ and eel migration disruption. And the last is we would like to thank to the Ministry of Finance, Republic of Indonesia through the LPDP Rispro Invitasi Program 2020 for contributing to this research funding. Okay, next. Thank you. That's all my presentation. Thank you for your attention. Assalamualaikum warahmatullahi wabarakatuh. I give back to Pak Irham as moderator. Thank you, Pak.
Thank you, Bapak Octaviano. What? I don't know. Octaviano. Yeah. Pak Octavian, we have only 1 more minute to discuss about your finding. So if anybody have some questions, please raise your hand and then ask the questions. All right, I don't think that somebody has some question because they are preparing for the presentation. So the next speaker will be Ibu Rosi Rahayu. Are you here, Ibu Rosi Rahayu? Okay. It's too dark. I don't know. You have to have a light so we can see your face.
Backlight.
Okay.
Okay.
All right. Thank you very much. So time is yours, Ibu Rosi. Welcome.
Okay. Thank you, Mr. Muhammad Irham, has been giving me the opportunity to present my presentation. Assalamualaikum warahmatullahi wabarakatuh. Hello, good afternoon, everyone. Today, I'm going to present my presentation. The title is Assessing Horseshoe Crab Avoidance Response of Flicker and Continuous Red Light LED for gillnet bycatch mitigation. The first, I want to introduce myself. My name is Rosi Rahayu. I'm lecturer in fishing technology in Department of Utilization of Fisheries Resource at Shakwala University. Okay, let me begin the presentation by explaining the background of the research. Next. Okay, let's start by looking at this figure. Okay, in the figure is the bottom gill net. Bottom gill net is the fishing gear used to catch demersal fish. One of demersal fish that being main target is a blue swimming crab. But in fact, not only the main target that caught by the gill net, but another bycatch too, it's called as a bycatch. Bycatch is an issue, both ecologically and economically. Moreover, if the bycatch is the protected animal, it would be harder to handle it. One of the bycatch that protected by the government is the horseshoe crab. Horseshoe crab is protected by the government because it's an ancient species and it's rare. Actually, the horseshoe crab has many advantages for us. The blue blood used as a serum to our medicine, drugs, and our vaccine too. But unfortunately, the horseshoe crab is a problem in the fisheries. If the bycatch caught by the gill net, it would be hard to release. So that to saving time, our fishermen forced to rip net off so that there is technical damage, and our fishermen suffering like money because there is additional charges to repair their fishing gear. Next. The same problem happened in Subang and in Mayangan. Mayangan is West Java province in Indonesia. And Supat Miningsih said that there are 46% horseshoe crab dominated in the total catch. So this is a problem for the fishermen. So that the study about mitigation by catch of the horseshoe crab is needed. The one solution that we can use is the visual acuity. Why the visual acuity? Because in the daily activity, horseshoe crab dominantly use the visual, like spawning, migration, and finding food. So that the technology that we can use is the LED. Next. Next.
I think we have technical issue right now. Suddenly we have a technical issue, so please be.
Yeah. Belum, Kak. Kak, kayak mana nih, Kak? Udah hilang itu.
Oh, terus. Okay. Okay.
Okay, we can see that the LED has many times used as the technology for mitigation by catch, such as the turtle or bird. And the previous study, the most horseshoe crab avoid the red light LED because the horseshoe crab prefer the short length wave than the long length wave, like a red color. And the short length wave is like a green or blue or the violet. And considering Rahayu 2019 that has compared many intensities, she found that the more the horseshoe crab exposure by the continuous light LED, the more horseshoe crabs show the aggressivity. And if it apply in the field directly, so the potency of the horseshoe crab tangle is still quite high. So that next, to prevent that happens, we use the flicker LED to comparison to continuous. Next. And based on the problem, the study aims to compare horseshoe crab response to continuous LED and the flicker. And the second one is determine the best red LED light to reduce horseshoe crab bycatch. And this study has benefit too as the technology and the solution for the government and the fishermen about horseshoe crab bycatch mitigation. And there is hypothesis too, there is difference of horseshoe crab responses when horseshoe crab avoid the light with different LED. Next. Okay, this study was conducted on February till August. It's so such a long time. In the 2019, the place Fish Behavior Laboratory in Bogor Agriculture University, and the method used was experimental laboratory. Next. And this is the tools and material that we use to this research. There is a bloodworm, it's a food for the horseshoe crab, and sand and the seawater, and the infrared to tracking the movement of the horseshoe crab. Next. And this purpose, the best on, and the as for the analysis, based on the purpose. The purpose one is to compare pattern and time of response between the flicker light and continuous. And for the pattern and time, the data that we need is video tracking of horseshoe crab movement. And for the pattern of response is analyzed with descriptive, and time of response is analyzed with normality test and with the independent test. Next. And for it, the purpose to determine which the best red light LED between a continuous and a flicker, we can compare it. Which the best based on the criteria. Next. To determine which the best criteria of the pattern of the response, the criteria is divided into three. That is avoid the light, it's marked with SD or LD pattern, and when it approach light, it's marked with SL or DL, and if no response, it will be stagnant. And the best criteria is SD or LD. Next. And this is the result. This is the result of comparison of horseshoe crab response between red light continuous and the flicker. On the upside is the response of horseshoe crab when avoid the light. And yeah, when it's the below is when horseshoe crab approach the light. Between the two types of LED, the flicker is the best. It's the better than continuous because there is no horseshoe crab that approach the light, but in the continuous does. Next. And this is the responses of horseshoe crab to the flicker based on criteria. There is approach, stagnant, and the response of avoid. Although there is no horseshoe crab approach,
Ibu Rosi, you have run out of the time right now, so.
Oh, okay.
We have to stop yours. So because we have another speaker. Thank you very much. Give Ibu Rosi applause. All right. For the next speaker will be Professor Khaliluddin. Are you here?
Yes, Pak. I am present, Pak Khalil.
Okay. All right. Time is yours. So start your presentation now.
Okay. Thanks, moderator. And thanks you for the opportunity. My name is Maya Yuliana from Department of Fisheries Resource Utilization, Faculty of Marine and Fisheries. I will present about sustainability analysis of pelagic fisheries using porsen at Ujung Serangga Fishing Port, Aceh, Indonesia. Next. Introduction. According to Basari 2019, the porsen is a very effective fishing tool for catching pelagic fish. The continuous use of fishing result in effective fishing, resulting in the depletion of fish resources. Warren 2021 and HISO 2021. For this result, good governance is needing as a requirement for sustainable natural resource management. Equity 2021 and Hilbo et al. 2021. Therefore, result on the sustainability of pelagic fisheries is important to explain and at the same time find solution in maintaining the sustainability of pelagic fisheries again porsen. This study aims to determine the sustainability of pelagic fisheries with porsen fishing to determine the sustainability of pelagic fishing with porsen fishing grid based on ecological, economy, social, and technological dimension. Purpose. The purpose of the study was to determine the sustainability of pelagic fisheries and the most influential factor on the sustainability of pelagic fisheries. Next. Time and result location. This result was conducted in February 2021 at fishing port of Ujung Serangga, Southwest Aceh District. The complex result location is present in Picture 1 below. Picture 1, result location. Next. Method of collecting data. The method used in this result is descriptive quantitative methods with survey and interview techniques. The data used in this study consists of primary and secondary data. Primary data is data obtained from interview and for interview with respondents who argued by questionnaire or attribute that have been prepared previously. Secondary data is data obtained from the Department of Relevant Agency as well from libraries relevant to research. Secondary data used in this study is data on the number of porsen fishing gear. Next, data analysis. The analysis used in this study is the rapid appraisal for fisheries analysis, which is a fishery rapid assessment analysis rapid. Analysis included RAP analysis, leverage analysis, and Monte Carlo analysis. The diagram of the two describe to sustainability with sustainability status in a multi-dimensional way, namely by comparing the performance of each dimension. Figure 2, initiative view for rapid analysis version 3.0. Next, result. Table 1, sustainability index category for each system dimension under study. Number 1, dimension ecological index value 68,33 with sustainability category were sustainable. Number 2, dimension economical index value 48,86 with sustainability category less sustainable. Number 3, dimension social index value 77,43 with sustainability category highly sustainable. Number 4, dimension technological index value 68,69 with sustainability category very sustainable. Figure 3, diagram of index analysis sustainability status of sluggish fisheries. It can be seen in the key diagram. The most influential dimension is the economy dimension with the score 48,86 with sustainability category less sustainable. Next, attribute sensitive. Figure 4, attribute that give a dominant influence of the sustainability of sluggish fisheries is by cat with the score 12.40, then source of working capital with the score 11.15, then government assistance with the score 13.72, and then fisherman education level with the score 7.28, then pre-sales processing with the score 9.62. Next. Conclusion, sustainability of sluggish fisheries with around trawler fishing equipment in PPI Ujung Serangga on the ecological dimension is at a sufficient level of sustainability with a score of 68.33. The economy dimension is at a level of less sustainability with a score of 48.86. The social dimension is a sustainable level with a score of 77.43. While the technology dimension is a fairly sustainable level with a score of 57.02. Attribute that give a dominant influence of the sustainability of sluggish fisheries with the red trawler fishing equipment is a ecology dimension. The bycatch of which is with a score 12.40. Economy dimension, assistance for the government with a score of 18.27. And attribute of working capital shows with a score of 15.182. Social dimension, fisherman education with the score of 7.28, and technology dimension, resource management with a score of 9.62. Next. And thank you.
All right, thank you very much. Give her applause. Our next speaker will be Professor Khaliluddin also. So the talk is about analysis of diversity index. So who is presented to Professor Khaliluddin?
Thank you very much, Mr. Irham.
Okay, time is yours and welcome.
Thank you very much, Mr. Irham. My name is Mandariski Purnama. I am master in Department of University for Fisheries and Faculty of Marine and Fisheries, University Kuala. Analysis of diversity index, dominant and composition of bags stand in Masjid Raya Water, Aceh Besar, District Indonesia. Next. Troll in triple of troll, there is to catfish, rock pelagis, and the marsal wind are the bigs. This fishing gear of the fish troll in active by seeking the skull of fishing nest. The cat of bag target of Dagang and Duyung are for people boot in not yet. Their the study was determined the generator of bags bank in Dagang in Royal field Dagang into the sick composition of the cat. Next. Next. Research methods session 1 Dagang in session 2 Rio. Next. There three of the base median of the fishing grade session 1 150 X session 2 203 meters. There's composition of the cat during the study include fish species session 1 60X and section 2 3 through 8 species. The his cat by now fish session 1 Chariskidai and session 2 Chariskidai. Diversity on diversity session 1 and session 2 session 1 minus 1.9 and session 2 1.3. Diversity index, the diversity index of session 1 and session 2 1.9 and session 2 minus 1.3. Then dominancy index on soft spoke the column. One is 0.0x in session 1 and 0.39 in session 2. Okay, thank you. Thank you very much. Next, thank you.
All right, thank you very much. Give him applause. So we have 6 more minutes to discuss about it. So if you have question, please raise your hand or about the problem. Okay. You don't have any more question. We're gonna have another speaker. It will be also Professor Khaliluddin about the macrobenthos. So anyone presenting for Professor Khaliluddin?
Yes, I'm here.
Okay. Okay, you are welcome. And then time is yours.
Thank you to the moderator. My name is Sela Yusnita. I'm from Department of Fisheries Research Utilization, Faculty of Marine and Fisheries, Universitas Syah Kuala. I will present about macrobenthos as an indicator of water quality assessment in Samudra Fishing Port Kutaraja, Aceh, Indonesia. Next. Introduce. The development of Samudra Fishing Port Kutaraja, Banda Aceh is quite significant, marked by many activities, both in the form of marine such as fishing port and activities around the coast such as settlement, industry, business, and aquaculture. The high activity in Samudra Fishing Port Kutaraja area has an impact on the environment, one of which is the decline in water quality at the port. Muningar et al. 2017. Due to the large number of activities at the port, it is suspected that water quality will decline. Changes in water quality in the port can be seen from the biological parameter based on the structure of the aquatic community of organisms that act as by indicators. One of the aquatic organisms that can be used as an indicator of water quality is benthos, because of its sedentary nature. Chunha et al. 2019. Benthos is one of the efforts to monitor the effect of port activities on the environmental status of marine water. Based on this description, this research is considered necessary to determine the type of benthos found in the harbor water environment so that the environmental quality of the Kutaraja Samudra Fisheries Port can be seen in order to embody a clean and pollution-free fishing port. Purpose. The purpose of this study is to determine the quality of the water environment seen from the benthic community structure, namely density, diversity index, uniformity, and dominance index, to embody the clean and pollution-free Samudra Fishing Port Kutaraja. Next. Material and method. This research was conducted in February 2021 for a month at Samudra Fishing Port Kutaraja or PPS, Banda Aceh, Aceh. This research was carried out using a quantitative descriptive method, specifically by interpreting the data or sample that had been collected. Then the sample were identified for the type of each species. Bento sampling was carried out for 1 month with an interval of 7 days of observation at 5 stations. Next. Data analysis. The parameter analyzed include the calculation of density, diversity index, uniformity index, and dominance index as follow. Number 1, density. Number 2, diversity index, where each smaller than 1 low or unstable diversity. Each greater than 1 and smaller than 3, moderate or less stable diversity. Each greater than 3, high or stable diversity. Number 3, uniformity index, where uniformity index value ranges from 0 to 1. If the value of E is greater than 0.6, then the uniformity value is high. Number 4, dominance index, where the value of the dominance index ranges between 0 and 1. If the value of C is close to 0, it means that almost no individual dominates. While if C is close to 1, it means that there are individuals dominating the population. Next. Result. Based on the table above, the quality of the water environment at Samudra Fishing Port Kutaraja from Station 1 to Station 5 has almost the same value and does not have a significant difference with a moderate diversity value, or each greater than 1, which means that the aquatic environment of Samudra Fishing Port Kutaraja is in the less stable category, but it has a high uniformity of benthic individuals, or E greater than 0.6, which means that the individuals are evenly distributed and low dominance value, or C is close to 0 or not more than 0.3, which means that there are not dominant individuals because the benthic fauna can survive and have the characteristic of high tolerance, which is an organism that is often found in declining water. Next. The difference in the value of the ecological index occurs because of the bento species that have a higher number of individuals found at each station, including Nereis diversicolor, Anodontia edentula, Nasarius pullus, Polynices didaima, Polynices sp, and Macropiotric belly. Nereis diversicolor was found at Station 1 because it liked muddy substrate. At Station 2, the most common benthic species is Nasarius sp because this species likes sandy or muddy sand substrate. Station 4, which is located close to the river mouth and there are mangrove around it. The bento species that is more commonly found is Anodontia edentula. Can also be found is muddy substrate such as at Station 5. At Station 5, Nasarius pullus was found as the dominant species on muddy sand substrate and very abundant in mudflat. Station of 5, benthic type Polynices sp and Polynices didaima are also commonly found because these species like muddy substrate. In addition, Macropiotric belly or snaking star are also found at Station 5, which is an area that has muddy substrate. This condition occurs due to activities at Samudra Fishing Port Kutaraja, such as water from washing kettles that flow into and into water ponds, disposal of fishery mine, loading and unloading ships, washing ships, fishing activities at the port, community walls, and other activities in the water. Conclusion. The water quality at Samudra Fishing Port Kutaraja is included in the moderate diversity category, which means that the water environment of Samudra Fishing Port Kutaraja is less stable. Okay, thank you for all attention.
Okay, Thank you very much. Give applause to the representing Professor Khaliluddin. We have 3 more oral speaker and 4 more oral speaker, and we're gonna have 7 poster. So we're gonna end up the meeting at 4, and then we're gonna have Salat Asar performing, and then after that we're gonna go again after the Asar. So the next speaker will be Bapak Adrian Domora. So he will presenting about the potential of spiny lobster fishery in Aceh Coast water. So time is your Pak Domora, and welcome.
Okay, thank you, Dr. Irham. I will share my presentation directly. Assalamualaikum warahmatullahi wabarakatuh. First of all, I want to introduce myself. I am Adrian Damora from Faculty of Marine and Fisheries, Syahkuala University in Banda Aceh. For this good opportunity, I would like to present our mini review entitled The Potential of Spiny Lobster Fishery in Aceh Coastal Waters. I wrote this article with Dr. Nur Fadli from Syahkuala University, Dr. Sabto Andriono from Airlangga University, Dr. Duranda Kembaren, and Professor Agus Suman from Research Institute for Marine Fisheries. Okay, the honorable participants, there are some issues regarding the spiny lobster fishery in Indonesia, especially in Aceh. There are, in 2017, the contribution of Aceh fisheries export to the national fisheries export was 0.30%, and the spiny lobsters made it to 5 largest contributors of this sector. And almost 100% of spiny lobster production in Aceh comes from the capture fisheries. In 2016, the Minister of Marine Affairs and Fisheries of Republic of Indonesia released a regulation of Capman number 56/2016 concerning the prohibition of fishing and or releasing spiny lobster from Indonesia unless it meets several conditions. This regulation then becomes a pros and cons among lobster seed companies because it is considered to help kill their export business to several countries in Asia Pacific. Then in 2020, the new minister revoked the regulation and replaced it with Permen KP number 12/2020 that the export of spiny lobster seeds is allowed to be exported. This regulation is also controversial because it is claimed not to support the sustainable use of spiny lobster resources. But in 2021, the new Minister of Marine Affairs and Fisheries reissued Permen KP number 17/2021 concerning the management of spiny lobster in Indonesia, which prohibits the capture and trade of spiny lobster seeds from Indonesia waters. So this review aims to discuss scientific information about the spiny lobster fishery from previous studies, especially research conducted in the southern western coast of Aceh, and also estimating their stock status and sustainability. The honorable participant, all spiny lobster species in Indonesia waters can be found in Aceh coastal waters. There are lobster pasir or Panilurus homarus, lobster batu or Panilurus penicillatus, lobster bambu or Panilurus versicolor, lobster mutiara or Panilurus ornatus, lobster Pakistan or Panulirus polyphagus, and also lobster batik or Panilurus longipes and Panilurus longipes femurus tiga. The last two species of lobster batik I have never found, but have been reported in other studies. Regarding to the distribution of spiny lobster in the nature, each spiny lobster species has its own habitat preferences. Lobster batu and lobster batik are distributed in rocks. Lobster bambu is distributed in coral reefs. Lobster pasir and lobster mutiara are distributed in sandy areas, and lobster Pakistan is distributed in the sand muddy areas. Then related to the catch composition of spiny lobster in Aceh, lobster batu, lobster pasir, and lobster batik dominate the catch of fishers. Lobster bambu, lobster mutiara, and lobster Pakistan are caught in small quantities, but lobster mutiara generally give the highest price. How about this fishing season in relation to seasonal trends? Lobster pasir is mostly caught during the dry season or southwest wind and high waves from May to July, where the other species lobster species experience a significant decrease in the catch. And how about the stock status of spiny lobster? In Indonesia, there are fisheries managed using the fisheries management area approach. And every year, Ministry of Marine Affairs and Fisheries uses the stock status for each fishery commodity. The status display in the form of flag shift, when the green indicates the fish stock is still good, yellow indicates the fish stock has been fully exploited, and red indicates the fish stock has been overfishing or biologically unsustainable stock. Spiny lobster stocks in fisheries management area or FMA 571 and 572, including Aceh coastal waters, has been overfishing, or we known as biologically unsustainable stock. And by modeling the surplus production model with the scaver model, we have mapped the status of spiny lobster stock from 2007 to 2016, and the results show the stock of spiny lobster is in FMA 571 and 572 has been in overfishing and overfished condition since 2008. For the note, overfishing and overfish status does not mean the lobster cannot be caught, but the fishing must be controlled. And how about the catch potential of spiny lobster in this FMA? Aceh has the maximum sustainable yield, or MSY, of spiny lobster about 292.09 tons per year, and the total allowable catch about 233.92 tons per year. MSY and also TAC are management measures in fisheries that can be used to ensure the sustainable fisheries. So the last for my presentation, to achieve the management goal of spiny lobster fishery, all stakeholders also need to ensure the eco-friendly fishing practices in the spiny lobster fishing. And the second one, do the critical coastal habitat conservation and also threatened species conservation. And the last but not least, stakeholder also do the socio-economic sustainability practices for the community. Thank you very much for your attention, and terima kasih. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam. Thank you, Pak Damora. Well presented by Pak Damora. So we have 2 minutes for discussion and questions with Pak Damora. If you or anybody in this room have some question, please raise your hand and talk and ask about the spiny lobster interesting topic actually. So you have any question, please raise your hand. I'm sorry, Pak Damora, there is no question for you. So
okay, thank you, Pak Irham.
Okay, have applause to Pak Damora. So our next speaker will be Neri Kautsari, Ibu Neri Kautsari from Samawa University. So are you here, Ibu Neri Neri Kautsari? Yeah. Neri Kautsari? No. Okay, we can keep for the last. So the next one will be Kurniawan Banjolu from Mosintu Institute.
Okay, ready?
Oh, okay, you are ready. Okay. Thank you very much for being here. Time is yours and welcome.
Okay, thank you very much for opportunity given to me. Now I want to introduce myself. My name is Kurniawan Banjolu. I'm from Mosintu Institute, Poso Central Sulawesi, Indonesia. And now I want to share about the checklist of endemic Adriantidae, Gobiidae, Senarcopteridae, and introduce fish in Lake Poso, Sulawesi, Indonesia. Next. Next. Okay, I want to share first one based on background and methods, result, and conclusion. Next. Okay. In Sulawesi, there are two ancient lakes, and Lake Poso, one of them. Currently, several endemic fish species of Lake Poso are thought to have become extinct. Since 1983, this endemic species, Adrianis iscruiti, has never been found. In addition, another endemic fish species has also been reported as rare, namely Mugilogobius amadi. Since 1985, the last report specimen of the species was in February 1987. Based on this, the impact of the introduction of the introduced species has occurred and is felt. So the number of species that are threatened with the degradation to extinction may continue to increase. In 2019, ichthyofauna in several central Sulawesi lakes, including Lake Poso, was reported, but the results have not been maximized due to limited observation station. Therefore, it is necessary to conduct research that aims to obtain the last information on endemic fish and introduced fish in Lake Poso. It's hoped that the result of this study, information on endemic fish and introduced fish in Lake Poso, will become on the base for the conservation and sustainable management of fish resource in Lake Poso. Next. Okay, the fish collection. The study was conducted in Lake Poso, Central Sulawesi, Indonesia from May 16 to 28 July 2019. Sampling was carried out at 17 stations, namely 12 at the lake shore for the inlet river and 1 station on the Lake Poso outlet. The fish sample obtained were separate from each station. The specimens were then prepared in 1 liter sample bottles containing 70% alcohol, labeled with the fish species name, collection location or station, collection date, and collector's name. The fish were then analyzed at the Animal Biosystematic and Evolution Laboratory, Department of Biology, Faculty of Mathematics and Natural Science, Tadulako University, Palu, Indonesia. Fish identification was carried out based on morphology character by Kotelat, Larson, Meissner, Parenti and Suroto, and Parenti, 2008. On the right of picture, you can see the map of distributing fish in Lake Poso. And this is 13 species we found in Lake Poso, 8 families, 11 genera, and 30 species, including 6 endemic and 7 alien fish. Next. This is the habitat of fish in Lake Poso. You can see 17 station sampling, and on the right you can see 8 of 17 sampling location with the water condition. There is clear water. Next. Okay, this Adriantidae, Gobidae, and Xenarcopteridae endemic to Lake Poso. Rono, Adrenitis oporus, one of 5 species Adriantidae in Lake Poso. And on the bottom, Nomorhampus celebensis, or julung-julung Sulawesi, or Poso half big. This is one of endemic species of julung-julung in the Lake Poso system. And on the right, you can see 2 color variation, male and female of Mogilogobus arsinorum, or we call bungo. Next. This is introduced cichlid in Lake Poso. So very adaptable, grow, and these species very potential to be invasive species in Lake Poso. Next. This is 32 species in Lake Poso, but on the study we found just 13 species. Next. Okay. This is the 10 of endemic fish in Lake Poso. In this study we found 6, and you can see the status by IUCN. The decline of endemic fish population: 1, introduce alien fish; 2, disease and parasite; 3, pollution and overfishing; 4, use of unfriendly fishing gear and habitat change. Next. Okay. Conclusion. The first, 6 endemic fish are still found in Lake Poso: Adrianitis oporus, Orisias nigrimas, Orisias orthognathus, Mogilogobus arsinorum, and Nomorhampus celebensis. And 7 species introduced fish were found in Lake Poso: Anabas testudinius, Aplocelus pancax, Shana striata, Cyclasoma or fronthorn, Melanochromis auratus, Oreochromis niloticus, and Trichopodus trichopterus. The existence of cichlids, fronthorn or Cyclasoma sp, Melanochromis auratus, and Orisias niloticus has the potential invasive fish in Lake Poso because of their adaptability and reproduction. Several conservation strategies for endemic fish in Lake Poso are protecting lake against the introduction alien species and essential habitat or in situ conservation, to protecting the habitats, regulation of fishing gear and fishing season, as well as the areas, save the areas, ex situ conservation and preparation of local wisdom. This various conservation we must be serious concern from the government, academics, and the community to save the spectacular endemic fish and ancient lake, Poso Lake, from extinction. Next. Okay, I want to thank to our colleagues and thank you very much.
All right, thank you very much, Mr. Kurniawan P. Banjolu. Thank you. So our last speaker that present today is Bapak Said Afdal Ar-Rahimi. Are you here, Said Afdal? Oh yeah, you are here. Okay, so time is your and welcome.
Okay. Assalamualaikum warahmatullahi wabarakatuh. Mr. Irham, thank you. Can you hear my sound?
Yeah, clear.
Okay, clearly. Thank you. I would like to present our scientific articles and maybe summary or abstract here. By title, Feeding Preference of Herbivorous Fish, Family Scaridae.
Pak Afdal, can you make a full screen?
Oh yeah, okay. I'm sorry. Myself, Afdal Rahimi, Mr. Ega and Mr. Andik Isdianto and Mr. Oktias Muzaki Lutfi contributed for this article by title Feeding Preference of Herbivorous Fish Family Scaridae. As we know, parrotfish is one of dominant species in Indo-Pacific, especially in coral reef ecosystem areas, which have 9 genera and 83 species. And in ecological perspective, the feeding behavior of this fish, parrotfish, or Indonesian call is kakak tua, is very important due to control of population of algae in coral ecosystem. And so we now relate to about coral recovery or coral resilience. They use their jaw, these species, to excavate and scrape algae with living on coral substrata. So sometimes resulted on addition and substrate and coral. Most parrotfish or kakak tua species forage by biting algae that grow on the surface of that coral. And this makes the coral clean for some time to provide substrate for other living species. Along with this process, the substrate or reef where the algae live is also often eaten by parrotfish. The aim of this research to record the diet preference of scarid or Scaridae family fish in Nature Conservation Foundation Putri Menjangan Nature Conservation Forum Putri Menjangan in Bali, located in Bali Island or in Indonesia, as water inhabited by parrotfish in its coral ecosystem. The research was carried out from January 2020 to February at the NCF Putri Menjangan. The location of the research can we see after this page in the figure 1. Observation of the observation point are considered to represent the coral condition of the water for the observation station. After defining the point of observation, then determining the coordinate using GPS, the research station determined in reef flat and slope areas with depth between 5 and 10 meters. So we collect the data at 5 meters for shallow and 10 meters deeper. The point of the research station can be seen, Putri Menjangan, NCF CF or Nature Conservation Forum at north of Buleleng location in Bali. This is the Putri Menjangan station. We can see in picture. The growth of scarlet fish were recorded by underwater camera. So we record about 1 minute. The activity by scarlet were calculated after that manually and photographed. So we collect the data from the video recording every 1 minute. In this picture, you can see number of bites from many species of Scarus niger, Chlorurus blackberry, and other species of Scaridae. You can see the at first station or number 1. Scarus koi, the highest number of our prevention of bite per minute. The result of study showed that 10 species from 2 genera, we find the scarlet species, and we found Chlorurus and Scarus, the 2 genera only. Was dominantly herbivorous fish in this area. The highest number of fish bite at station 1 or the first station was obtained from Scarus koi species with a total 41.27 bite per minute. While station 2 and 3 we obtained it from Scarus rivulatus species with a number of 40.96 bite per minute and 41.59 bite per minute respectively. So the majority of parrot species found choose to forage on dead coral with algae, we call DCA, substrate, because the water conditions were not optimal for the growth of the coral. When water condition is not optimal, usually grow the algae more aggressively. So many coral died and the skeleton were overgrown by algae. In this picture, you can see some species from Chlorurus blackberry, Oedema, Sordidus female and Sordidus male, and Roselle and Scarus altipinis. And these pictures, Scarus dimidiatus female, Scarus dimidiatus male, and Scarus papipectoralis, and Scarus niger, Scarus koi, and Scarus rivulatus. Thank you, Mr. Ilham.
Thank you very much, Pak Said Abdal. Thank you. That is the last oral speaking.
Welcome.
For today. So we are going to have 7 or 8 more poster presentations. So I just want to ask question to the forum, to this room of meeting, whether we continue for seven more speakers, or we stop and then praying and then continue for 7 more speakers. So my suggestion is, because of we still have, sorry from the people outside of the Aceh. Aceh still have very high sun right now, so we are having 20 more minutes of Asar, so I think it's good enough. I think it have a time enough to have 7 more speakers. So I think we are going to continue for the poster presentation for 7 more presenters. So are you here all? Ibu Salmarika, is Ibu Salmarika here? Okay, and then Bapak Muhammad Arif, are you here, Bapak Muhammad Arif? And Ibu Imelda Agustina, are you here, Imelda Agustina? Yeah, okay. And then Bapak Adli Waliul Perdana, are you here? Yeah. And then Bapak Edi Miswar. Okay. And then Bapak Rian Juanda. Okay, I think all of them are here, so it's better for us to continue, and then we can later on we stop the our meeting. So for the first poster presentation, please welcome Ibu Salmarika. So time is you, Ibu Salmarika.
Yes, I am. Oke, Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everybody. Before I start, I will say thank you for Mr. Irham because give me the good opportunities to stand here. Now, I will try describe our study that title performance analysis of catch fisheries in Sabang City. And as we know, Sabang City is one of the center of capture fisheries production and the tip Sumatra Island, which has a large potential of fish resource. Yes, so we must keep those fisheries sustainable. The sustainability of fisheries fish resource can be maintained by good fisheries management. Fisheries management in practice must be observed the dynamic of fishing activity like fishing gear and fishing area, fishing flat, and technology by fishermen. And based on the fact, fisheries management should be carried out with available accurate data and information so that the output product is excellent and on the target. So this study can be important to do. Furthermore, the research was conducted from May to July 2021 in Sabang City, and effort to collect data in this study were conducted by survey method and data analyze, and present in form of picture and narrative. And then the study result are the production of capture fisheries in Sabang City is dominant by large pelagic such as tuna, skipjack, and bullet tuna. And then fishing activity in Sabang City are classified into small scale or traditional, and the dominant fishing gear is hindline, and fishing flat dominate by fishing measure 0 to 5 person, and fishermen carry on catching area usually around the coast or fast not 2 nautical mile from fishing base to Bruh Island or Ras Island and Aceh Island. I think that's all Thank you for attention. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam. Thank you, Ibu Salmarika. So the next presentation will be Mr. Muhammad Arif. We are welcome you, Mr. Muhammad Arif. So time is yours.
Thank you, Mr. Irham. Thanks for Mr. Irham. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone. Let me introduce to myself. My name is Muhammad Arif, and I am lecturer at Department of Utilization of Fisheries Research. Today, I will present my research. The title is Development of Fishing Vessel Shipyard in Traditional Lampulo, Banda Aceh. My abstract is the research was conducted the types and criteria of wood used as raw material for traditional shipbuilding, and now the process of traditional shipbuilding. The method used in the study is a descriptive method with interview technique and direct observation in the field. This research is located in the Kutaraja Ocean Fisheries Port, or PPS, Banda Aceh. The result show that the type of wood commonly used as raw material for shipbuilding in traditional shipyard were bingkarai wood, laban wood, bala wood, meranti wood, tembusu wood, bungur wood, and merbau wood. Meanwhile, the traditional shipbuilding process generally began with laying the keel and installing the crest. Next, continued by frames installation, hull plank, then upper hull main frame. Hereinafter, deck floor installation, hatches manufacture, and superstructures construction. The final stage of shipbuilding process is matching, painting, installing engine, propeller, and steering. Introduction. The study aims to determine the types and criteria of wood used as raw material for traditional shipbuilding, and now the process of traditional shipbuilding. Research and method. A descriptive method was used in this research using interview technique and direct observation in the field. Research in discussion. Traditional shipbuilding process. The first shipbuilding process at the Lampulo traditional shipyards is determining the dimension and sketches the ship owner will make. After that, the selection of quality wood material or block is carried out according to the ship part to be built. After the preparation stage is complete, furthermore process is of cutting the wood according to profile. Types of wood used. The types of wood can be seen in Table 1. The difference in the use of wood species as raw material for traditional shipbuilding show the diversity in the use of plant species. It is closely related to the social cultural condition of each region and the availability of natural resources. Equipment use. Some of the equipment used in traditional shipyards in Lampulo can be seen in Table 2. The result of field observation rarely found modern electronic equipment. Nevertheless, the ship produced in this region have proven their capabilities strength. Conclusion. The results show that the types of wood commonly used as raw material for shipbuilding in the traditional shipyard were bangkai wood, laban wood, meranti merah wood, tembusu wood, bungur wood, and merbau wood. Meanwhile, the traditional shipbuilding process generally began with laying the keel and installing the crest. Next, continue by frame installation, hull plan, then upper hull main frame. Hereinafter, deck floor installation, hatches manufacture, and superstructures construction. The final stage of shipbuilding process in matching, painting, installation engine, propeller, and steering. Thank you, Mr. Irham.
Okay, thank you very much. The next presenter will be Ibu Imelda Agustina. Please welcome Ibu Imelda Agustina, and time is yours.
Okay, thank you, Mr. Irham. Hello, everyone. Good afternoon. My name is Imelda Agustina. Today, I'm going to present my paper. The title is Coral Reef Fisheries Development Business Strategy in Sabang City. Introduced, marine fisheries are important source of food and income for people over the world. 1 billion people, many around developing countries, rely on fish as their main animal protein source. Fisheries provide an important safety net for people when all their sources of development are unavailable or following natural disasters. Learning on this potential, the fisheries business in Indonesia shows a very good picture. Fisheries provide nets that are essential to people when other world resources are unavailable. The aim of this study is the strategy for developing coral fisheries in Sabang City. Okay, the research methods are time and place. This research was carried out in Sabang City, Aceh Province in May 2021. Sabang City became a place for coral reef fisheries business development. Data collecting method. Primary data were collected through direct observation and in-depth interviews at location with the dominant intensity of reef fishing activities by filling out the prepared questionnaire. Secondary data collection was carried out through literature studies, internet, journals, and Fisheries and Marine Affairs Department of Sabang City with issues relevant to research topic. The data collected are developmental data, types of fish, appropriate fishing units. Then the data is analyzed by using SWOT analysis. In the SWOT analysis, there are internal and external factors. By that factors, we will create an opportunity and threat strategy. The result from research fisheries development strategy in Sabang City. In the internal strength factor that is obtained, there is a strategy. Strength and opportunity. 1. Increase reef fish production results. And 2. Improving reef fish price quality to be export and factor. Strength and threat is 1. Developing fishing gear technology to replace non-environmentally friendly fishing gear. 2. Conduct socialization on coral reef conservation where reef is life. And then factor external, there is a strategy, weakness, and opportunities. 1. Expanding access to social network of fishermen group with outsiders. And 2. Conducting outreach activities, outreach to regular assistance in care of coastal village in Sabang. Strength and threats. 1. Cooperating with community in superfishing fishing areas by utilizing local culture, which is still very strong in protecting the sea to avoid illegal and excessive exploitation. And then the latter is the conclusion. The result from development of coral reef fisheries in Sabang City. is one, coral fishing technology development in Sabang. Two, increase supervision of fishing areas for fishing gear that is not environmentally friendly will damage fishing grounds. Three, transportation and distribution for reef fish trade with high selling value. And then four, that quality capture fisheries product development for fishermen's welfare income. I think that's all the presentation of my paper. Thank you.
All right, next speaker will be, thank you Ibu Imelda. Next speaker will be Bapak Adli Waliul Perdana. Please welcome Pak Adli Waliul Perdana, and time is yours.
Yep, thank you Mr. Iram for your present. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon ladies and gentlemen. Let me introduce myself. My name is Adli Waliul Perdana. I'm from Department of Aquaculture, Faculty of Marine and Fisheries, Universitas Syah Kuala. Thank you for joining to this presentation, and honors to meet you in this beautiful day. I would like to share about analysis of distribution patterns and marketing margins of capture fishery products at the Ujung Baru Fish Landing Base, West Aceh District. I did this research with my team. Okay. Distribution patterns of capture fishery product at PPI Ujung Baru has never been reported. Besides that, many parties participate in marketing activities, so it's necessary to study to the distribution pattern to see if the distribution pattern at PPI Ujung Baru is appropriate, beneficial for all parties, especially fishermen. The objective to be achieved in this study are to determine the distribution pattern of capture fisheries product at PPI Ujung Baru and determine an efficient distribution patterns in conducting a marketing system by knowing the margin received. The method used is in this study is a survey method with the purpose of sampling data collection technique. The data analysis used in the research is descriptive analysis and marketing margin analysis. The analysis used to describe the distribution pattern in PPI Ujung Baru. Marketing margin analysis is used to measure the profit of each actor involved in the catch distribution patterns. Results of study obtained to distribution patterns that occur in PPI Ujung Baru. The first distribution pattern is fishermen to bench stock, to wholesaler, to retailers, and to customers. Second, fishermen to bench stock, to trader, to consumers. Both distribution patterns at PPI Ujung Baru are correct because these patterns get a marketing margin value of 50%, and the fisherman profit sharing value of 50%, where the distribution pattern is classified as efficient. The party who benefits from good distribution patterns is Bentoke because they get a seller's profit of 7%. So this is my presentation today. I apologize if there is a mistake. Thank you for listening to my presentation. I hope what I have shared will be beneficial for all of us. Thank you.
Thank you, Bapak Adli Waliul Perdana. So the next speaker will be Pak Edi Miswar. So are you here, Pak Edi Miswar?
Yes, I am here.
Oh, are you presenting for Pak Edi Miswar? Okay, so time is yours. Welcome.
Okay. Assalamualaikum warahmatullahi wabarakatuh. Hello, everyone. Firstly, thank to Master of Ceremony for giving me the time to speak in front of you all. And honorable to all of listeners, let me introduce myself. My name is Novi Erianti. I come from Department Fisheries Research Utilization, Faculty of Marine and Fisheries, Syah Kuala University. I would like to present of my journal under the title Management Status of Belut Tuna of Usk Room with EAFM Approach Pusong Fish Landing Base of Semarang City. The water of Semarang City are one of the potential fishing area on the east coast of Aceh. The catch in this area is generally landed at the fish landing base or Pangkalan Pendaratan Ikan PPI Pusong Lok Semawe City. The fishing gear that is commonly used is fursen, which catches large pelagic fish and small pelagic fish, one of which is bullet tuna. The production of bullet tuna cast in 2013 until 2015 increases, but decreases in 2016. Then from 2017 until 2018, it increases again, but decreases in 2019. Therefore, it is necessary to evaluate the management of bullet tuna at PPI Pusong Lok Semawe City. Management assessment was conducted based on EFM criteria. Of the 6 EFF domain, 1 domain was chosen to be focus of the assessment, namely the fish research domain. Then, the result of the study is to specify indicator of the fish research domain on the management of EIFM-based bullet tuna fisheries and to assess the direction of EIFM-based bullet tuna fisheries on fish research domain. Indicator in the Pusong Lok Semawe City landing base. The research conducted this research in February 2021 in PPI Pusong Lok Semawe City. The method of data collection was through the survey. The data analysis used the data from each indicator of fish research domain, then processes by tabulation and analysis by simple composite with arithmetic average and display in fact model. Okay. Ecosystem approach to fisheries management or EIFM is sustainable fisheries so that the utilization of fishing can utilize in the long term by considering the ecosystem. The fish research domain consists of 6 domains, namely catch per unit at 4, trend, then besides 3, the proportion of juvenile cow, 4, composition of cat, 5, rank colleagues, and 6, endangered, threatened, and protected species. Then the result of the assessment of all fish research domain, the composite value of the fish research domain is 68.3, or a good category with a green color model flag. So that the management status of bullet tuna or auxiliary as PPI Pusong is the good category. For this CPUA graphic and then fish size, frequency of bullet tuna at PPI Pusong, then composition of percent catch at PPI Pusong, you can see at the figure. I think this is enough my presentation today. Thank you for your attention. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you, representing for for Mr. Edi Miswar. So the next speaker will be Bapak Rian Juanda. Is anybody? Oh, Bapak Rian Juanda is here. Welcome, Bapak Rian Juanda. Time is yours.
Okay, thank you, Dr. Muhammad Irham. Maybe I won't take 1 minute to her Azad. Okay. Okay. Thank you, Dr. Muhammad Irham. Good evening. Good afternoon, everyone. Okay, I would like to present my poster by title Study of Groupers Renidae landed in the traditional fishing port in Kota Bawah Timur, Kecamatan Sukakarya, Sabang, Indonesia. Okay, Sabang City is one of the most remote area in Western Sumatra, which has a larger potential for capture fisheries resource. The location of the city of Sabang, which is now as a coastal area, is located between two waters, namely the Indian Ocean and the water of the Malacca Strait. Fishermen in the coastal area of Sabang City generally use fishing rods. So this study aims to determine the composition of the grouper's catch, Renidae, consisting of types of grouper fish, the number and size of individual groupers landed at the fish landing base of Kota Bawah Timur. Okay. Sampling was carried out as much as 10% of the total catch on 3 February until 3 March 2020. Data analysis by identifying the catch, the composition of the tip of fish, count, and the morphometric size of the catch. The result showed that there were 8 types of grouper fish, namely the red sun groupers, Cephalopolis miniata, and then tomato groupers, Cephalopolis sonerati, tiger groupers at Pinephelus ciplitocepeps, and then rubber sunu groupers at Pinephelus fasciatus, and then blackmouth groupers or Atelperca roga, and then scissor groupers, Variola albimarginata, and then black sprinchex grouper, Cephalopolis argus, and branch grouper, Variola lauti. Okay. You can see in the result too, in picture two, catch and fishing gear picture. Okay, there is a type of groupers. With a total catch of 1,038 individual in the Sabang. The most common species were Cephalopolis miniata. Okay, you can look at the pic in result too. Okay. That the fish Icfaes Fkp species using hand fishing rod and fishing rod. So in the Sabang, catching and fishing gear by fish arrow and fishing rod. Okay, thank you, Dr. Irham.
All right, thank you, Bapak Rian Juanda. You have 2 presentation actually, so you have another one, right?
Sorry, the next is blank.
Okay, all right. So the last presentation will be Professor Muhammadan. So he was absent for the beginning. Now he is representing by siapa tadi? His student. So we are welcome Professor Muhammadan to present the presentation. So time is your Muhammadan.
Yes, I hear.
Okay.
Thanks to the moderator. Assalamualaikum warahmatullahi wabarakatuh. My name is Zara Nur. I went to present a feature article poster with the title of my research is Study of Amino Acid Contained in Seahorse Hippocampus sp.
Would you make a full screen, please?
Seahorse Hippocampus sp is one of the high value marine fisher commodities. This study aims to determine the value of the amino acid contained in Seahorse Hippocampus sp. This research took place in March 2021. The method used in this study was the OC Association of Official Analytic Chemistry method, which used the so-called apparatus for the proximate gas and the GCMS gas chromatography mass spectroscopy apparatus for the amino acid test. Sampling was carried out in Simelu water and carried out at the Environmental Quality Testing Engineering Laboratory Unit, Faculty of Engineering, and at the Agriculture Product Analysis Laboratory, Faculty of Agriculture, Shakwala University. The analysis used is proximate analysis and amino acid analysis. The proximate content of Seahorse is water content 29%, as on 25.43%, fat not content 0.57%, protein content 32%, and carbohydrate 48.48%. The amino acid composition of large seahorse and small seahorse consists four types of essential amino acid, namely leucine, isoleucine, histidine, and valine, and 6 types of non-essential amino acid, namely alanine, glycine, asparagine, arginine, and serine. Thanks from the explanation of my paper. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you for the presenting today. So, the share screen. Can you close the share screen, please? Can you close the screen? Hilang. Share screen-nya ditutup sebentar, tolong. All right. So that is the end of our presentation in our room 3. So thank you very much for the presenting the research, the very best research of today in the fisheries and environments. And all of the speakers, we thank you very much for your contribution to our conference. So we will meet next meeting. I think next year we'll have one meeting like this also, but I don't know whether it's online or offline. So I don't want to talk more about the thing because time is running out. So I want to close everything and thank you very much for the attention. We are sorry. We apologize if we have something under control. So because the technical issue and something like that. Thank you very much and see you next. Assalamualaikum warahmatullahi wabarakatuh. Oh, let me tell you something. Before we leave, can you show your face? Because we want to take some pictures. Okay, so your video and now we're gonna have pictures. Everybody, please, especially the speakers. Okay, Ibu Salmarita, are you ready to take pictures? Okay, do it. Okay, everybody is smiling, please. The big smile is the good one. Okay, thank you very much. See you next year. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Pak Irham.
Thank you, Dr. Irham. See you later.
See you together or see you later? All right, I'm going to leave the meeting right now. Thank you.